Alright, hello everyone. So it's Miss Ivy, your instructor, and this is chapter 16. It is the last chapter for our semester. So everybody give yourself a pat on the back for getting all the way to the end. So chapter 16 is going to segue away from microevolution and discuss evolution on a large scale, which is called macroevolution. Oh, hold on a second. Let me swap the sides. There we go. Okay, so here's an outline for this chapter. It has 3 sections. We'll talk about what macroevolution is and relate it to the concept of speciation. We'll talk about the fossil record and finally systematics. So like we mentioned in chapter 15, we talked about microevolution. We discussed how natural selection promotes changes in allele frequencies over a relatively short amount of time. These changes can accumulate in a population and lead to macroevolution, which is evolution on a large scale, where these changes in a population occur over a very long period of time, which can lead into a concept called speciation. And speciation is the splitting of one species into 2 species or more due to changes in these allele frequencies within our gene pool. So big idea here is our microevolution can accumulate into macroevolution, which can then lead to speciation. So what's a species? So in order for us to really understand this concept of macroevolution, which is again leads into the divergence of species, we need to know what in the world a species is. So by definition, the biological species concept says that members of a species always interbreed. All right, they interbreed. Members of a species breed together. What that also means is that one member of a certain species cannot breed with a member of a different species. So for example, cats and dogs, they don't breed together because they are not the same species. So members of a species interbreed. Cats breed with cats, dogs breed with dogs. So because they interbreed, they must have a shared gene pool. And finally, each species is reproductively isolated from every other species in some way. And we'll talk about some mechanisms of reproductive isolation in a little bit. But again, all that leads back into is cats breed with cats. They don't breed with any other species. Dogs only breed with dogs. Humans can only breed with humans. Species number 1 of whatever species of bird will only breed with the same species of birds. For example, flamingos won't breed with ostriches. Okay. And another thing about our biological species concept to remember is it's not based on appearance. So, for example, flycatchers, they're a group of birds that look very similar, but they're different species. And so these different species of flycatchers do not breed together because, again, by definition, a species only breeds amongst the same species. Another example of where appearance is not based on the biological species concept is with humans. We all look very, very different from one another, with the exception of, identical twins, but we do breed together. We're all still the same species, even though we may look very different. So appearance has nothing to do necessarily with species. You cannot base it on that. What you base species off of is whether or not they can breed together. So here's an example of what we were referring to, the flycatchers. This is flycatcher number 1, 2, and 3. They look really similar, but guess what? They are not the same species. So if you look down here at the bottom, it gives you this the scientific name here, here, and here. They're all in the same genus. If you recall way back from chapter 1, this is the scientific name, the genus and species. They have the same genus, but they're not the same species. So thus, they do not interbreed. And again, humans, we look very, very different, but we are the same species and we can interbreed. So like I mentioned a couple minutes ago, we said we would talk about some reproductive barriers. How is it that these three species of birds here can't breed together even though they look really, really similar? Why is that? All right, so let's talk about those things. Those are reproductive barriers. What prevents organisms from interbreeding across species? So there's several different isolation mechanisms that prevents reproduction across species. The first set are known as pre-zygotic barriers. And if we break that down, pre means before, and zygotic, you can think of the term zygote. And we talked about zygotes previously when we were talking about meiosis. Remember, remember, zygotes are the very first cell of a new individual. So pre-zygotic means that we are preventing reproduction before the zygote is even formed. So no zygote is formed. There are several different ways that pre-zygotic barriers can occur. Habitat isolation, temporal isolation, behavioral isolation, mechanical isolation, and gametic isolation. And we'll talk about each one of those. But I just want to give you a list for now. The second main way that we can prevent reproduction across different species is post-zygotic reproductive barriers. Post meaning after, and of course zygotic referring to the zygotes. That means that there's some kind of reproductive barrier that occurs after the zygote forms. So sperm meets egg, but something happens after that zygote forms that causes isolation within species when it comes to reproducing. And those three things, and again we'll talk about them in detail in a few minutes, are zygote mortality, hybrid sterility, and F2 fitness. So we'll start going into more detail first with our pre-zygotic reproductive barriers, and then we'll talk about the post-zygotic ones afterwards. So pre-zygotic isolation, we're going to start with habitat isolation. Habitat means, where something lives. So habitat isolation means that two species live in different habitats. And because they live in different habitats, they're not going to encounter one another very often. And therefore, if they're not encountering one another, well, they're obviously then not going to make babies together. So that is our first example of a type of pre-zygotic isolation. Pre-zygotic, again, meaning zygote never formed. In this case, zygote doesn't form because the two different organisms never come in contact with each other because they live in a different habitat. So, for example, garter snakes. These are two different garter snakes here. One is a water dweller and the other one lives on land. Well, because one lives on land and one lives on the water, they don't ever meet up, so they don't ever try to reproduce together. So they don't share a gene pool, separate species. Likewise, we have some crickets here. These crickets, they're in the same genus, as you can see, Gryllus, but they are different species. They do not interbreed. They have this is another example of a habitat isolation because this guy here prefers sandy soil, whereas this one here prefers more of a loamy soil. So because, again, they have different preferences for their habitat, they're not going to hang out together, they're not going to reproduce together. All right, so that is habitat isolation. Our second type of pre-zygotic isolation, which again means no zygote, is temporal isolation. And so the way I like to think of this is think of like tempo. So if you're familiar with music, the tempo of the music is, how fast it's playing. So in this case, temporal isolation is a type of prezygotic barrier where two species breed at different times of the day or different times of the year, and so they don't attempt to mate. So here's one great example here. We have these two frog species. They're different species, but they reproduce during different times of the year. So because of that, they don't interbreed, and they're different species. Secondly, we have two different types of skunks over here. They're the same genus, but different species. The one on the left here, they breed during the summer, and this guy here is going to breed during the winter. So because, again, they breed at different times, that's an example of temporal isolation, which is a prezygotic barrier that prevents reproduction across different species. And this is not selective to just animals like the frog and the skunk. This happens in plants too. So certain plants will release pollen during certain times of the year, and others will release their pollen or whatever reproductive means that they have during different times of the year. So just like that with animals, we see the same kind of thing with plants and other different types of organisms. But all in all, that's called temporal isolation, tempo, time. Our third example of a type of prezygotic isolation is behavioral isolation. So with behavioral isolation, organisms of certain species have different behaviors that attract their mates that would be unattractive to the opposite sex of a different species. So this picture here demonstrates two different species of birds. This guy or gal, whatever it is, male, female, is singing a song. And the members of the same species would find that song attractive. But this bird of a different species says, ugh, I don't like that song. So that's an example of behavioral isolation. Another example, and I like this one because I feel like they stick out. So these guys here, they're called blue-footed boobies, and they have these really bright, pretty blue feet. And it is a attractive trait for reproduction in their species. And I want to show you a couple of videos up here that demonstrates a couple of these behavioral isolation courtship patterns that we see in birds. So let me see if I can pull it over. Alright, so y'all get the idea here. So this is a very specific behavioral thing that occurs specifically in this species. If this bird would have shown its blue feet to a cardinal, the cardinal would be like, what are you doing? That's weird. Get away from me, because it is not attractive to them. All right, so that is behavioral isolation. I have another one I want to show you guys because I think it's kind of funny and kind of cute.
And called the superb bird of paradise is a magician with a special trick. First, he advertises his show by calling to attract a female. And then, with the audience in place, he begins to work his magic. That's impressive. But she needs to step out for a moment to think. Oh boy. Well, not today.
Okay, so I just thought that one was very interesting. But anyway, it's just another example of behavioral isolation. So, if this blue-footed booby were to, come in contact with that other bird, I'm not sure what kind that one was, that had that elaborate dance that it was doing, it would not be thinking of mating with it because it's not their behavioral courtship patterns. So these behavioral isolation mechanisms or tactics are just another example of prezygotic isolation. They will not reproduce or attempt to mate across species because they have different courtship patterns. All right, so the next type of prezygotic isolation is mechanical isolation. And so when you think of mechanical, you think of parts of something. And that's exactly what this is about. Mechanical isolation in this sense is when our animal genitalia or even plant floral structures are just not compatible and reproduction can't occur. It's another example of prezygotic isolation. So this means zygote never forms because the genitalia just don't fit. So as we can see here, I thought this was kind of funny, but there's a giraffe attempting to mate with a donkey, but that is not going to work. It's not going to go over well because their genitalia do not correspond with one another's. Another example would be here. We see mechanical isolation of the genitalia between these two different types of snails. These two snails here are different species. Remember, it's not about how they look because they look really similar, but they are not the same species. Because of that, their genitalia openings are not going to align whenever they're in their mating position. You can see genitalia 1 and 2. They're not aligned. And so, goodness, what that translates into is that mating can't occur because the genitalia don't fit together correctly. And some more examples are shown here. These are different genital structures in male damselflies. Those are kind of like dragonflies. You can see every single one of these looks very different, pretty different from each other. This is species 1, 2, 3, and 4, and they are not identical. And because of that, they are going to cause mechanical isolation where they can only reproduce specifically with females in their same species. So mechanical isolation, genitalia or plant floral structures are not compatible. Next is gamete isolation. So with gametic isolation, the organisms of the two different species are allowed to let's see, their reproductive structures may come in contact with one another, but the gametes won't fuse to form a zygote. So this can happen for a few different reasons. For example, maybe the sperm can't survive the reproductive tract. Maybe it's too acidic, or it has certain types of chemicals or bacteria within the female reproductive tract that incompatible sperm just can't survive in. Sometimes eggs may have receptors on them, protein receptors. Remember we talked about those several chapters back. So there's protein receptors that are found on cells. In this case, the egg cell, it may have a receptor that's specific to sperm of a specific species. So those are just another examples of prezygotic isolation. However, in this case, sperm might be able to get really close to egg, but they won't fuse to form a zygote. So a great example, real life example, would be sea urchins. So we see here on the left a purple sea urchin and a red sea urchin. They are of a separate species, even though they look really similar. But the sperm of our purple sea urchin cannot fertilize the eggs of our red sea urchin. The reason for that is the sperm cannot survive the reproductive tract of our red sea urchin. And so because of that, they're never able to make it to the egg to penetrate to form the zygote. So again, that's another, our last example of pre-zygotic isolation. Okay, so that summarizes our pre-zygotic isolation reproductive barriers. So now let's talk about post-zygotic. Post-zygotic means the zygote does form. So sperm meets egg, but something may happen after that that causes reproductive isolation between species. The first example of post-zygotic isolation is zygote mortality, and so that one's kind of, obvious. The zygote is not viable, meaning that that the zygote dies, typically before birth or very, very soon after birth. This is usually due to a chromosome incompatibility, where you may have different numbers of chromosomes. The chromosomes from mom versus dad can be giving instructions to the body plan that are saying two different things, and it just doesn't work out. So, for example, sheep and goats, they can mate together. You see them here hanging out. Look how they love each other. It's so sweet. But if they attempt to reproduce, a zygote will form, but that zygote does not develop properly and will result in miscarriage. Over here, we can see a hybrid individuals develop, potentially between this tiger and leopard. Whenever that occurs, the zygote begins to develop, but the pregnancy ends in miscarriage or stillborn. The zygote, again, the chromosomes are not compatible. They may be set, the instructions in the DNA are telling the cells to do two separate things. It just doesn't work out. So that again is an example where an individual will not be able to produce a viable offspring. But again, this is post-zygotic. They are able to mate. Sperm fuses with egg. We have a zygote, but after the zygote is formed, something happens that causes the zygote to not be viable. That is zygote mortality. Zygote death. Death. The next example of post-zygotic isolation is hybrid sterility, and lastly, F2 fitness. Hybrid sterility means that a hybrid zygote does develop and grows into an adult, but it is sterile as an adult. A great example of that, especially y'all may know around here, living in a rural farming community. If a horse breeds with this donkey here, they can mate. They do produce a viable offspring, which means it lives. It's called a mule. But this mule usually cannot reproduce. So that is another example of post-zygotic isolation. Zygote was formed. In this case, we do have adult mules. They live and everything's all good, but that mule will be sterile. They will not be able to continue a gene pool because they are sterile. They cannot breed. Now, in rare occasions, mules are capable of reproduction, but in those rare cases, their offspring, which would be the F2 generation, have never been able to reproduce. We've never had any examples of that to date. They do not form viable gametes in the F2 generation. So after our horse and donkey breeds, we get our mule. That's our offspring, F1 hybrid. If you recall, from Mendelian inheritance, this would be the P generation, this would be the F1 generation. We call this a hybrid because it is a mix between two different species, horse and donkey. Then in the F2 generation, if this mule is capable of reproduction, which is not common, then that offspring in the F2 generation cannot reproduce. They don't form viable gametes. So that puts a halt in the gene pool, post-zygotic isolation. All right, so those are our examples of reproductive barriers that ensures that members of the same species reproduce together and interbreed, and that members of one species do not interbreed with members of another species. Okay, so now that we understand why species, not sure what the plural there is, but now we understand why members of these species are only going to interbreed with the members of the same species. So now let's talk about some models of speciation. So the first type of model that we're going to discuss is allopatric speciation, and it's based on geographic isolation. And again, we said speciation is divergence of one species into two or more. With allopatric speciation, this speciation is due to a geographic barrier. Now that could be mountain ranges, it could be an ocean or a lake, or a volcano erupted and created a valley due to all the lava. It could be I'm trying to think of some more examples. Ooh, here's a good one. A comet hit the Earth and it created a large crater separating individuals of the same species for a long period of time. All right, so that's just a couple of examples. All those are geographic. Okay, so think about geography, the state of the Earth. Excuse me. So let's look at an example that's shown in our textbook. So this picture here, of course, is the state of California. And these are several different pictures of various species of salamanders that are located in California, as well as where they're located. So this guy here, all the species or members of this species are located in this yellow area. In the green, we have this species. In the blue, we have this one, and so on and so forth. Now, there was an ancestral population in the Northwest that were the common ancestors to all of these different species. And the Central Valley within California separates the populations that have descended over many generations of time. So that limited contact allowed genetic changes to accumulate, again, microevolution, over time to a point where interbreeding was very, very rare and now impossible amongst these different species. So again, we started off as this one, and here's my salamander, don't make fun of my drawing. This one salamander species was the ancestor, located again Northwest California. But as they migrated across California and were exposed to different geographical barriers, they stopped interbreeding, populations stopped interbreeding amongst each other. And over long periods of time, genetic mutations accumulated. And now if we were to take this guy here and transplant him over here into this area, he would not breed with this other one. They're members of separate species. There's some kind of reproductive barrier that prevents them from breeding because they have been separated for so long that there's some type of genetic changes that causes them to no longer be able to reproduce. Our second example is sympatric. So sympatric speciation is where a population will develop into two or more reproductively isolated groups without geographic isolation. No geographic isolation is required for this type of speciation. This is particularly found in plants due to what we call polyploidy. And polyploidy is where we have multiple chromosomes. If you'll recall from previous chapters, we talked about euploidy, aneuploidy, polyploidy, many chromosomes. So, and this type of speciation is actually how we got bread wheat. You know, the stuff we're common that we have today. Is where we had a cross between two different types of wild wheat, as you can see here and here. It produced a sterile hybrid, but something that's common in plants is they like to duplicate their chromosomes. We don't talk about that a lot in Bio 1, but in Bio 2 we do. But that doubling of the number of chromosomes is how we got breadwheat. And they can reproduce with other polyploids or any other members of a similar organism with the same number of chromosomes. So that's sympatric. No geographic isolation is required for that type. All right, our third type of speciation that we'll discuss is adaptive radiation, and this is where we have a single ancestral species that gives rise to many different species in a very short amount of time. So whenever I hear the term adaptive radiation, I kind of like to think of the sun, because the sun emits radiation. And what I think about is, here's my ancestral species. I'm going to write AS for ancestral species. And all the sun's rays are the many species that ancestral species gave rise to. And the reason why all these different species came about is because they are adapting, hence the term adaptive radiation, to their specific niches or environments. So a great example that we've talked about in the past, we didn't call it this back then, but it was the Galapagos Island finches. We said there's tons of different species of finches, and they all had very specific niches where they would be adapted to that certain niche, where they would eat a certain type of food in a certain type of area. We had our land dwellers, our tree dwellers, some that finches that ate very large nuts and seeds, some that were adapted to eating insects and all that good stuff. So we have lots of different finches that have evolved from a single ancestral species based on them adapting to specific environments. Excuse me. Now, the reason why for all of this is because, well, our populations on these different islands in the Galapagos were subjected to being forced to become adapted to certain niches due to founder effects, genetic drift, mutations, and just all in all the process of natural selection. But each particular population became adapted to a specific habitat on its island, a specific food, a specific locale. So all of these new finch species arose, and they do not interbreed. They are not capable of reproducing across species. All right, and here is just some of the species that that are part of the Galapagos finches that we had mentioned in previous chapters. So you see our tree finches, and again, this is not an inclusive picture. There's many more. There's hundreds of species of finches. And our ground ones, you see the ones that are on the ground, they tend to have larger, very powerful beaks for crushing stuff, mostly seeds and nuts. And you have the other end of the spectrum over here and up at the top. All right, so that summarizes the things that are in 6.1. In 6.2, we talk about the fossil record. And the fossil record is really important because we can trace the remains from past life for, a very, very long time in the past. And it gives evidence of, what kind of organisms were around in the past. We are able to date these fossils and kind of trace the lineages of organisms, or at least have evidence for it, in a very large period of time. This field is called paleontology. So these are scientists that discover and study fossils. They study the fossil record. They make decisions about the history of species. The next concept we'll discuss is the geological time scale. And so, like we mentioned before, these fossils can be dated and estimated where they are placed in the geological time scale. And geological time is huge. So, you think about our daily lives, we have 24 hours in a day, 365 days in a year, and, our lifespan's about, 80 years, if we're lucky. But humans have only been around on Earth for 0.04% of the history of life. We haven't been here that long compared to how old the Earth is. So let's look at some examples of fossils. Here we see a leaf fern. This leaf fern is from about 245 million years ago. It was buried in some sediment and hardened into rock. We have this here, which is it's called a midge. It looks kind of like, sort of like a mosquito. It's an insect from 40 million years ago, embedded into some amber, sort of like tree sap. And lastly, we can see a fossil from 47 million years ago that is evidence of a early insectivore, which is something that eats insects, but it is a mammal. But so those are just 3 examples of some fossils. But fossils are any remains from past life. It can be an impression in sediment. It can be defecation, poop. It can be bones, etc. Any remains from past life. So where do we find these fossils? We typically are going to find fossils, as pretending we're a paleontologist, in the strata, which means the layers of rock of the Earth's crust. Typically, the oldest fossils are going to be deeper, and the ones that are younger will be more towards the surface. But as we study fossils in the rock layers, it can enable scientists to construct a geological timescale that includes, what organisms existed in approximately what time periods. And this geological time is huge. Like I said, we've only been around for 0.04%. But our geological time extends for a very long time, 4.6 billion years, as a matter of fact. 4.6 billion years is the estimated age of our Earth. This 4.6 billion years is divided by the geological timescale into eras and periods and epochs, and we'll take a look at what those mean in just a moment. And there's one I didn't mention on here, but also eons. So eons are the largest geological time unit. Our textbook doesn't include eons in the picture that I'm about to show you, so you should be able to view this picture in your textbook as well. But it does start with eras. So, and I'm just going to show you really quickly the next slide, because there's a continuation of this. It's very big. All right, so we're going to actually start with this slide, the second one, because this is further back. As you can see, I'm gonna move back to the previous picture. This is the earliest, or it's not earliest, but the most recent time in geological time. So you can see, here we are, humans. So let's go back. Let's go back all the way to the beginning of time. So about 4.6 billion years ago. So again, this is, go back again. This is in millions of years. 4,570 million years ago is the same thing as saying 4.6 billion years ago. That's when we estimate that Earth formed. Now, I want to make sure y'all understand that you do not need to memorize anything on this chart, okay? I will tell you what I want you to know. I want you to be able to read this chart, but I do not require you to memorize anything on it unless it is on the study guide. Okay, so our first prokaryotic cells begin to form about 3.5 billion years ago. We see a huge fluctuation in oxygen accumulation in the atmosphere. We talk about that a lot in bio too, but that is essentially one thing we believe that enabled eukaryotic organisms to begin to exist, to form. Our protists then diversify. We begin seeing soft-bodied invertebrates like jellyfish after that. All these things occurred during what we call Precambrian time. And Precambrian time is 87% of the geological time scale. There's little to no oxygen during this time period for the most part. There's no ozone to shield all the radiation coming in from the sun. So we see very, very basic organisms during this time period. Now, there's lots of things that happen between Precambrian time and the next era, which is going to begin with our Paleozoic era. For example, we already mentioned O2 accumulation. There's several other things that occur. We won't really get into that in Bio 1. But basically, the atmosphere in the Earth starts to change. And that is believed to have led to a explosion of different types of phyla emerging on the Earth. So we begin to see marine algae, very basic invertebrate phyla become present. And as we move up and up towards more recent time periods and eras, we see more and more complex organisms as we get to our modern day time. All right, so at the very top, this is the most recent part of our geological time scale where we see the age of the Homo sapiens, which is us. All right, so just as a reminder, again, we mentioned the terms era, period, and epoch. Again, those are just sections of time. So kind of think of it like, if we are discussing our watch, our clock, we have hours, we have minutes, and we have seconds. Seconds are very short. We have minutes, which are, a little bit longer, 60 seconds in a minute. And then we have hours, which there's 60 minutes with an hour, etc. So they're just, they're units of time. That's what eras are, and that's what periods are, and that's what epochs are, as well as eons. Eons are very, very long periods of time. This picture doesn't even represent those, but it does represent eras, periods, and epochs. So our most current epoch that we are in right now, which is from 0.01 million years ago to present, that's the age of Homo sapiens. That's right here, right now. Our previous epochs, and again, I don't require you to memorize these. I don't even require you to be able to pronounce them because some of them can trip you up. But the Pleistocene, Pliocene, Miocene, Oligocene, Eocene, Paleocene. All of these different epochs fall within the Quaternary period and the Tertiary period. So this would be Quaternary, and Tertiary. And within these and these periods, Quaternary and Tertiary, they're all going to fall into the Cenozoic era. So again, you do not need to memorize that, but you do need to understand how to read the chart. So if I were to say, I would show you this picture, and I would say, what era did the first hominids appear? Well, you can see over here, the first hominids is listed there. Well, if I asked you for the era, it would be the Cenozoic era. If I asked you for the epoch that the first hominids appeared, you would tell me the Pliocene. Okay, so that's what my expectations are for this, for the geological time scale. Our next era is the Mesozoic. That's further back. We have within the Mesozoic era our Cretaceous, Jurassic, and Triassic periods. This is, was largely, I guess, associated with dinosaurs most commonly because, y'all know, I know y'all have seen Jurassic Park and all that good stuff. But a lot of other things were going on during this time. So you can read those here. I won't bore you to death by reading them to you, so you can do that on your own. Moving further back, the Paleozoic era includes the Devonian, Silurian, Ordovician, and the Cambrian periods. And then previous to that, again, we have Precambrian time. Okay, so that I know is a lot of information to take in at once. Again, you do not need to memorize any of that. Just just understand the chart. Know how to read it. Now, because it's so big, because life's history or Earth's history is so vast, it's oftentimes easier to visualize how much time is in geological time, 4.6 billion years, by placing it in reference to a single day. So if we were to do that, here is our clock. You see we have midnight is here. 1, 2, 3, 4, 5, blah, 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 all the way to noon, and then back to midnight. So this is one day if we go around this clock. If we compare geological time to a 24-hour day, Then prokaryotic organisms, which we said we have evidence for them existing 3.5 billion years ago. They would have existed at about 5 a.m. All right, so this is our oldest fossil records of prokaryotic organisms. I don't know why this is doing that. What? I don't know what button I'm hitting. I'm not hitting any buttons. This thing is so weird. There. Okay, prokaryotes appeared at 5 a.m. Our first eukaryotic organisms would have occurred at 4 p.m. That's right here. That's a long time for our first eukaryotic organisms to have appeared. Wow. All right, so again, at midnight, that's 4.6 billion years ago. Our oldest prokaryotes at 5 a.m. is around 3.5 billion years ago. Our first eukaryotes, which is around 2.5-ish billion years ago, would be at 4 p.m. So look at all this time that have passed, billions of years at this point. Multicellularity. We have our first records of cellular organisms at 8 p.m. Day's almost over. That's only a billion years ago. Only. Plants, 9 p.m. The dinosaurs existed at 10 p.m. And humans existed 30 seconds ago. Well, 10 p.m. was dinosaurs, and humans existed 30 seconds ago. If we were to take 4.6 billion years and shove it into 24 hours. So that's kind of amazing. Remember, I said that humans have only been around for 0.04% of Earth's history. It's pretty amazing. So hopefully that kind of helps you wrap your head around the vastness of 4.6 billion years. All right, so our next concept is the pace of speciation. Pace meaning, how fast something occurs. There's two different types of models that we're going to discuss for the pace of speciation. The first is our gradualistic model, and Darwin thought that evolution occurred very gradually, and what he meant by that is that we would see lots of intermediate stages if the gradualistic model held true. So for example, if we see this ancestral bird, we see it's kind of a pale orange, and its population was evolving due to geographic barriers or sympatric speciation, adaptive radiation, etc. We would see intermediates from what exists today. So here today we have the red coloration and the pale yellow. We see intermediates in the middle, where this pale orange bird diverged into a dark yellow, and then a light yellow, and then the lightest yellow, as well as another divergence of a darker orange to an even darker orange to a red. So that is our gradualistic model. There's gradual changes over time that results in intermediate stages found in the fossil record. But a lot of times these intermediate stages are not represented in the fossil record. So, does that mean the gradualistic model is inaccurate? No, not necessarily. It may mean that we just haven't found these fossils. There's just a lack of fossils that have been found so far. And it's difficult to indicate exactly when speciation has occurred because of all of these transitional links. So that's just one model. Our second and last model we'll discuss is punctuated equilibrium. So this type of pace of evolution discusses or encompasses periods of equilibrium where there's no change, but then that is punctuated or interrupted by abrupt speciation. And this model here, this picture, demonstrates what we mean by punctuated equilibrium. We have this ancestral species. There is a period of equilibrium. All of them look like this. this pale orange color. And then there's some sort of abrupt change that results in different lineages. Lineage 1 and lineage 2. This would be a common ancestor to this lineage and this one. So instead of seeing all these transitional links, we're not gonna really see that, because this was such a short, it was a punctuated, not gradualistic change. It's an abrupt change. So again, you don't see dark yellow to light yellow, or lighter yellow to an even lighter yellow in this lineage. You don't see the light orange to a darker orange to a darker orange to a red. It's orange, the orange bird with 3 different lineages that all end with either the light yellow, the orange, or the red, but there's no transitional links. There's no intermediates. That's the word I was looking for. All right, so transitional links are less likely to form fossils and less likely to be found with punctuated equilibrium. The differences between these two different models are subtle, but potentially important. So another thing to also take note of is when I say that they're subtle, the reason I say that there's subtle differences is because in this model, our punctuated equilibrium, we said that there's some kind of sudden change, but sudden in geological time can be thousands and thousands and thousands of years. Because again, geological time is so vast, 4.6 billion years. All right. So another thing we should discuss is the mass extinctions of species. So if you were paying attention to that picture that I showed you, the really big one, you might have noticed there's a couple things that talked about or indicated mass extinctions of species. And what a mass extinction is where we have a disappearance of a lot of different species in a relatively short amount of time. So there's 5 mass extinctions that are currently documented. We have one at the end of the Ordovician period, the Devonian period, the Permian period, the Triassic period, and the Cretaceous periods. And even today, we're seeing significant mammalian extinction at the end of our Pleistocene epoch. I'm going to go back to these images here so you can see what I'm referring to. So you can see significant mammalian extinction right here. We see mass extinction here, the end of the Cretaceous period. This is where 50% of all species went extinct. The dinosaurs, most reptiles went extinct. We have a mass extinction after the Triassic period. Lots of corals and ferns went extinct. We have a mass extinction after the Permian period. We call it the Great Dying, where 83% of all living species on land and in the sea went extinct. After the Devonian period, lots of coastal marine species lost. After the Ordovician period, over 57% of marine species went extinct. And we may be, very well experiencing that mass extinction occurrence right now, today. All right, so why do these things occur? Why does mass extinction happen? And there's lots of contributing factors, one of which that is well documented is continental drift. So you might have talked about this in another class, maybe like a physical science back in, I remember talking about this in middle school. But this is a theory where continents move on tectonic plates. And this can lead to movement of continents, very, very slow process, but it can lead to massive habitat changes. So this is just a picture here demonstrating the movement of tectonic plates. So for example, here's a plate here, here's a plate here, and they're moving apart. This can create a rift in the ocean. Think of things, it would cause like a trench. Think the Marianas Trench, if you've ever heard of that. If you haven't heard of it, it's really interesting. Deepest trench in the ocean. Weird stuff's down there. Some pretty cool stuff. But as these things are moving apart, they're going to be butting up against something else. So, for example, over here, as this plate is moving this way, it's butting up against this other plate. Well, when they start butting up together, something has to give. And what's going to happen is one's going to start moving underneath the other, and one's going to move on top. This can cause earthquakes. It can cause mountain ranges to form. It can cause volcanic eruptions, because it can affect what's going on in the mantle of the earth, underneath the earth's soil, in the subduction zone. It can cause volcanoes to erupt, which obviously can have a huge impact on habitat. So these movements of tectonic plates can definitely have an impact on habitat, and thus contribute to potential mass extinctions. So moving forward, the next concept is Pangea. So like we were just talking about, continental drift, continents move. We talked about tectonic plates. So what the Earth looks like today does not look anything near what we believe the Earth looked like a long time ago. I think 250 million years ago. In fact, this is what we anticipate the Earth looked like 250 million years ago. Instead of having multiple continents spread out across the Earth, we see one large clump of a landmass. We call this Pangea. All right, so here is Pangea. This is primitive Australia, Antarctica, India, Africa, South and North America, and Eurasia. They all were conjoined together. And over time, because again, land masses are divided onto tectonic plates which move, these land masses began to separate. So as we move forward, we see 65 million years ago, doesn't look anything like it did 250 million years ago. These tectonic plates moved, thus the land masses move, and now we see more of a similar Earth that we are familiar with. Still not quite there, but you see Antarctica is moving down here, and we have Africa and kind of where it is today, Eurasia, North America, South America, India, and Australia. Still moving, and I thought I had a picture of modern day Earth, but I don't. But we're probably pretty familiar with what our global map looks like. In fact, I'll pull one up. All right, so we can see what our global map looks like today. Okay. So, continental drift, is just one contributing factor that we believe leads to mass extinction. A second one is meteor impacts. So meteor impacts on the Earth, if they're large enough, can be similar to a worldwide atomic bomb explosion, where a mushroom cloud would block out the sun. Because the sun is blocked, that's going to lead to plant death, because as you know from our photosynthesis chapter, plants need sunlight. It causes temperatures to drop, and that can cause freezing. Not all animals or plants are compatible with freezing weather. It can disrupt food webs. If plants are dying, that's going to affect the herbivores. If our herbivores that are eating the plants are dying, then the animals that are eating those herbivores are not going to have a food source, and they'll die, and so on and so forth. This is believed to be the demise of the dinosaurs, this meteor impact. Now, why do we what evidence do we have for this? Well, there's Cretaceous clay that contains lots of iridium, which is not a very common element that's found in the Earth crust. Meteors, however, are very, very rich in this element. And there's a very rich layer of this iridium soot located near the Yucatan Peninsula. This is located in Mexico and is believed to be the site where a large meteor impact occurred that led to the demise of the dinosaurs. All right, so you guys, that's 16.2. We're going to now segue into 16.3, which discusses systematics and taxonomy. Luckily, this is going to be sort of a review for you guys because we talked about systematics and taxonomy way back in chapter 1. But we talked about how systematics, their goal is to study the evolutionary history of life. We talked about phylogeny before, which is where we group or classify organisms, and it's related to taxonomy. Taxonomy is where we identify, name, and classify organisms. Phylogeny is where we also take into consideration evolutionary history when doing this classification scheme. And this is all part of systematics. So another concept we talked about in chapter 1 that we'll review is our binomial naming system. Binomial means two names. Bi meaning two. Nome comes from the Latin word nomen, which means literally name. So we have two names. The two-name naming system is going to utilize the genus and the species or specific epithet. Do know that specific epithet is the same thing as species. So we will use the genus and the species or specific epithet, same thing, to name organisms. For example, Homo sapiens, genus, species, for humans. Canis lupus is for wolves. And this one's going to be a mouthful. Cypripedium aculae is the genus and species for this plant life here. All right, there is a couple of rules for naming organisms. Specifically, it's mostly about formatting rules. But always the genus is capitalized and the species name will be lowercase. So you can see in all of my examples, the genus is capitalized, species is lowercase. Capitalized, lowercase, capitalized, lowercase. Also, when you're typing, you always want to use italics for naming organisms. If you're writing it down, then it's standard to underline it. But this is just two parts of our bigger picture, our hierarchical system of naming organisms. So if we think back to our taxonomy hierarchy, we talked about this before. If you recall, domain, kingdom, phylum, class, order, family, genus, species. So hopefully you guys remember that. We had that nice little acronym that I gave you guys to help remember the order of most inclusive to least inclusive, where we have our most inclusive category at the top, which is domain. Domain eukarya includes all eukaryotic organisms, which are organisms that have cells that have a nucleus. And then we talked about kingdoms. We did phylum, class, order, family, genus, and then finally species, which is specific to only one species. Now, one category that is in this that we did not talk about in chapter 1 because it's relatively very new is supergroups. So not all textbooks even have supergroups listed in there yet. It's kind of, like we've talked about before, taxonomy is a changing field. We're always finding new organisms and finding new ways to classify them. And as those processes develop, sometimes we have new classes of animals come up, and in this case it's supergroups. So just something to know, this is an ever-changing field. All right, so our next concept is something we did not, that's not good. Story of my life, technical errors. It's always something. Okay. So, what was I saying? I want to switch this. Something that we didn't talk about in chapter 1, though, was phylogenetic trees. Actually, I think we did talk about them very briefly now that I think about it. But a phylogenetic tree is a diagram that indicates common ancestors and lineages or lines of descent. Common ancestors are always going to be or begin at the base of our phylogenetic tree. And remember, we said we call it a phylogenetic tree because it kind of looks like a tree where we have the trunk would be here, and then we have all these branches that branch off into other branches. So our common ancestor for all of these organisms that we see here, the sheep, the cattle, the deer, and the reindeer, existed way back here. And it's the order of Artiodactyla. So this common ancestor at the base of the tree has traits that are shared by all of these other groups within the tree. The order Artiodactyla, specifically, it has hooves with an even number of toes. So that includes our sheep, cow, reindeer, and our deer. Now, as this branches off, we see two lineages. We see the Bovidae and we see the Cervidae. That's going to be different families. We're getting more specific. Remember, kingdom, phylum, class, order, family, genus, species. We're getting more specific as we go up. Less inclusive. So this included all of them. Now we're only including certain ones. Bovidae are going to have, are not going to have antlers. They're going to include the sheep and the cattle. Whereas the Cervidae includes only organisms that do have antlers, such as the red deer and our reindeer. All right, so that is an example of a phylogenetic tree. We start off with a common ancestor that has characteristics that are inclusive to all of the lineages here. And as we get more specific, it excludes certain organisms. So again, Bovidae only includes these two guys. They don't have antlers. Whereas this family, Cervidae, includes only these two that do. All right. So how do we trace this phylogeny? Like, how do we come up with this? So we can use a lot of different data that help us to study these evolutionary relationships between species. Morphological data is one example. Morphology meaning physical traits. We talked about homologous structures in the past, so that would be a great example of morphological data that can be used to trace phylogeny and common descent. We talked about forelimbs and vertebrates. If you recall a chapter or two back, I had a picture that showed homologous structures amongst, I believe it was bats, and we had whales, horse, and human. But however, remember, how organisms look are not always the best data to have. I mean, yes, it can give us some insight, it gives us some evidence, but it can sometimes not be accurate, because sometimes organisms can look very similar even though they're not very closely related. And this is called convergent evolution. So up until now, we've been talking about divergent evolution, where organisms have a common ancestor and it will result in that ancestral population diverging, moving away into different species based on natural selection. Well, convergent evolutions is where we have traits that are acquired by organisms of a population that are seen in distantly related species. So a great example of this would be dolphins. So dolphins are not fish. They might live in the water. They have a dorsal fin. Sometimes people may think they're fish, but they're not. They're not fish. They're mammals. They produce milk. They have teeth. They breathe air. So those are things that are not always related with fish. That's just an example, one example of convergent evolution. They have, they have a fish-shaped body. Again, dorsal fins. They live in the ocean, but they are mammals. They're more related to humans like us, because we're mammals, than they are fish. They have these fish-like traits because they have adapted to an aquatic environment, but that does not mean that they are very closely related to fish. And that those examples that we gave, like the fish-shaped body and the dorsal fin, those would be examples of analogous structures, not homologous. Homology is due to common ancestry. Analogous structures have the same function, but no recent common ancestor is the cause of those analogous structures between dolphins and fish. Now, again, we said that morphological data, for that reason, is not always the most ideal evidence for phylogeny. Much more preferred is molecular data. Molecular data can include DNA sequences, RNA sequences, the protein amino acid chain sequences, the structure of our cellular machines like ribosomes, etc. Molecular data is a much stronger piece of evidence. Think of it like the gold standard for demonstrating evolutionary relatedness. So the more closely related species are, the more similar their DNA will be. Ribosomal RNA changes very, very little over across species. So that's a great reliable indicator that tells us how closely related species are. And to help us analyze all this data, as you can imagine, there's massive amounts of data that's available. Computer software has made it possible to analyze sequences of data, sequences of DNA, RNA, amino acids, very quickly, and very cheaply. This field of biology is called bioinformatics. I wish I would have got a degree in it because I feel like I could make a lot of money in it. But there's databases, they're available online. Look at PubMed if you're interested. The National Institutes of Health is the host, but PubMed, that have huge databases that are available to investigators across the world, with sequences of, thousands, hundreds of thousands, if not millions of different DNA, RNA, amino acid sequence for particular proteins for many, many species on the planet. So you'd be shocked at the immense amount of data you can find online. And this is really important though, because that means that investigators in science, we don't have to start from scratch anymore. We can find things online that other scientists have done and add to it ourselves with our own research. So, but just to give you an example of how we can analyze or interpret molecular data, this image here shows you a phylogenetic tree of various different hominid-like creatures and humans, of course. So here we have a galago, a capuchin, green monkey, the rhesus monkey, the gibbon, the chimpanzee, and the human. Well, if you're looking at this over here, where this shows you how many millions of years ago in the past, this common ancestor existed 40 million years ago. Lines up with the 40. This common ancestor is the common ancestor to all of these organisms. As we move to this location, anytime you see a cross here, it indicates a common ancestor. This is the common ancestor for the capuchin all the way over to the human. This is not the common ancestor to the galago. This is the common ancestor to the galago and the rest of these. Moving forward, if we look at this common ancestor, it is the common ancestor to the green monkey all the way over to the human. This is the common ancestor to the gibbon all the way over to the human. And this is the common ancestor to the chimpanzee and the human. Now, how can we interpret this? Well, Because the most recent common ancestor here is shared between chimpanzees and humans, humans are most closely related genetically and evolutionarily to the chimpanzee. We are more distantly then related to the gibbon, and even more distantly related to the rhesus and green monkey, and moving forward, the capuchin and the galago. So if I were to ask you, which of these organisms is the most distantly related to humans, you would tell me the galago. What if I were to ask you, what is the common, what two organisms is this the common ancestor for? Well, you see the two lineage are the green monkey and the rhesus monkey. All right, so that's how you interpret these phylogenetic trees. Moving forward into cladistics, I believe this is our last little bit here, but cladistics is just another way to trace evolutionary history by using shared traits that derive from a common ancestor to determine relationships. With cladistics, the goal is to create cladograms. These cladograms depict evolutionary history based on available data, which I know sounds kind of broad, so we'll go into a little bit more details. But essentially with a cladogram, you're going to have outgroups and in-groups. An outgroup is not part of the study, whereas the in-groups are part of the study. And this will start making sense once I show you some pictures. But any trait that is found in both the out and in groups is a shared ancestral trait, and therefore it's presumed to be present in a common ancestor. So let's look at an example. So when we construct a cladogram, we'll start off with a table. We're going to list several different organisms. We have at the top here the lancelet, which is an aquatic organism, an eel, also aquatic, the newt, a snake, and a lizard. And then we're going to list several different physical characteristics. So here we have the notochord and embryo. The notochord, you can kind of equate that to a primitive spinal cord, so sort of. The vertebrae, which is the backbone, lungs, three-chambered heart, internal fertilization, amniotic membrane within an egg, 4 bony limbs, and a long cylindrical body. So, and then we're going to determine which of these organisms have each one of those traits. So if it's colored in, that means it has it. If it doesn't have it, then it's not colored in. So our notochord in the embryo, all of these organisms have the notochord as an embryo. Vertebrae, the eel, newt, snake, and lizard have a vertebrae backbone, but the lancelet does not. Lungs and the three-chambered heart, that is only seen in these 3 guys, the lizard, snake, and newt, but not in the eel and the lancelet. Internal fertilization and the amniotic membrane, which would surround the egg, is only seen in snakes and lizards. The 4 bony limbs is seen in newt and lizard, and the long cylindrical body is seen in eels and snakes. So what does all that mean? This means as an outgroup, our outgroup is our lancelets, because they are not vertebrates. And again, vertebrates means they have a backbone. Our in-group, which are the members of the study. This particular cladogram then has 3 clades. 3 clades, right? Our vertebrate, our lungs, and three-chambered heart, which are together, and then internal fertilization with the amniotic membrane, right? So you can see they're color-coded. This is the notochord in the embryo is not going to be part of our study because all of the organisms have it, so it doesn't differentiate amongst anything. They all have it, but the remainder do. So again, vertebrae, lungs, and the three-chambered heart, which is in orange, and the ones in yellow. These are not going to be useful for us because if you can look at the pattern here, we're not. If they were only pink colored here and here, then that would be helpful. It would help us to narrow narrow our clade down, but because we're not seeing the same pattern, it doesn't. All right, so we only have 3 clades in this case because of that. All right, so if we take this chart, we can create a phylogenetic tree from it, where our common ancestor at the very, very back, which has all of the characteristics, which would be our lancelet. They have a notochord, they have the vertebrae, lung, all the stuff. Now, moving forward, the majority of the organisms here have a vertebrae. So you'll see they're going to be next on our phylogenetic tree. So vertebrates are here. That includes our eel, newt, snake, and lizard. As we get a little bit more specific, we have lung and 3-chambered heart development in this phylogenetic tree. That will include our newt, snakes, and lizards. And moving forward, we then see amniotic eggs with internal fertilization seen with only snakes and lizards. So this is how we construct and read a cladogram. So now let's talk about Linnaean classification versus cladistics. So remember Linnaean, oh my goodness. Linnaean classification, we talked about kingdom, phylum, class, order, family, genus, species. Here we're constructing phylogenetic trees based on traits. So, how do they differ? Our Linnaean classification system oftentimes doesn't coincide with how we would do things in cladistics. In Linnaean classification, for example, it places birds in their own group. But if we were to use cladistics, it would place them in a clade with crocodiles. Why? Because they both have a gizzard, they have scaly skin like other reptiles do. As a matter of fact, Linnaean classification is not consistent with new understandings of phylogenetic relationships. We still use it, but remember, taxonomy, cladistics, phylogeny, they're all a very vastly changing field. So some scientists are proponents of modifying our Linnaean classification system to make it better match cladistics and the molecular data that we are obtaining. Others say, no, let's construct an entirely different system. So I don't know what way the science community will go. I don't know when they'll make these changes or how fast it'll be, but it's very exciting. It's very a very dynamic field that is, like I said, very, very much changing and has been changing for a long time and will continue to change as we gain new information. So I don't know how old you guys are, but I know for me, back in the day, when I was learning about this, I remember, y'all probably won't believe it, I remember learning this stuff in I can't remember if it was like kindergarten or I think it was first grade. But the kingdoms that we talked about and the domains we talked about back then, this is 25 some odd years ago, it's not the same thing I'm teaching you today. It's similar, but it's not the same. And that's again because it's a changing field. All right, so however, right now at this moment, we still utilize our 3-domain system where we have our bacteria, archaea, and eukarya domains, and within our eukaryon domain, we have our 4 kingdoms, which we talked about way back in chapter 1, which is Protista, plants, fungi, and animals. So again, back in the day, it was different. We used to have a 5-kingdom system, which included plants, animals, fungi, and Monera, which is bacteria, but they've been moved and now have their own domain. So that's what I was talking about. 20 years ago, that's how I learned it, but now it's different. All right, so this is just a representation of our 3-domain system. You can see our bacteria, our archaea, and our eukaryotes with our different kingdoms within eukarya: protists, animals, fungi, and plants. And lastly, I know I mentioned this already, but just one more time I'll mention it again, our supergroups. So this is based on very current research. We are currently organizing existing kingdoms into supergroups, and this is the future of our of taxonomy, is including the supergroups that will connect newer evolutionary data based on molecular sequencing to help establish more accurate relationships between our eukaryotic organisms. All right, you guys, that was chapter 16. I hope y'all enjoyed our final chapter. I'm very excited that we are now at the end of the semester, and you guys hopefully have are very well versed in basic biology, and hopefully I hope it helps you in your future endeavors. I know lots of you guys are probably vet tech bound. Most of my students are. So I hope that you find that this information that you've learned this year is going to translate well into your science courses in the future. All right, you guys, have a good one.